Thank you. It's been a lovely day. I appreciate the opportunity to be here and to talk with you. I'm a pediatric oncologist. I take care of kids with brain cancer. And each week when I walk into clinic, the first thing I do is I sit down and I just pause and I say to myself, what am I going to do today that I don't want to be doing in 10 or 20 years? And as I go through clinic, I think about that, and we ask the question of, could we change that through the work that we do in our laboratory? And in 2004, we had a 17-year-old girl who had a brain scan that looked like this. You can see that there's something abnormal there, and we can tell that that's a tumor, but we really can't tell where it begins and where it ends. Now, if I were to give each of you a scalpel and ask you to cut out the brain tumor but leave the normal brain behind and don't hurt this girl, where would you start and where would you stop based on that MRI scan? Now, I could make it easier for you and take you into the operating room, and this is what you'd see. And we know that this is about where the tumor is, but we also know that this is the primary motor strip, that if you cut into that, she'll wake up paralyzed. So now if I gave you a scalpel, where would you start and where would you stop? It's very difficult. In this case, a neurosurgeon who was trained at Harvard, had 25 years of experience, was the head of neurosurgery, worked on this girl for 17 hours, and at the conclusion of that, he thought that he'd taken out the tumor and left the normal brain behind, but what we saw the following Wednesday, the day that I was in clinic, was that this big piece of cancer was left behind because with all the tools he had available to him in 2004, he couldn't tell that that wasn't normal brain and he didn't want to hurt the girl. So if these are tumor cells and any of those are resistant to chemotherapy or radiation therapy, this will grow and take her life. Or we could take her back out of the intensive care unit, back to the surgical suite, do another operation, place her at risk again, delay her chemotherapy and radiation. There is no good choice in these cases. So this particular day, Dr. Ellenbogen and I decided that we were going to create a way to make the cancer light up so that surgeons could very clearly see the cancer while they're operating. And we turned to a most unusual partner for this, partner in nature, the Israeli deathstalker scorpion. This scorpion happens to make many little mini proteins that are part of its venom. And there was a group down in Alabama that was studying these toxins and found one molecule that blocked chloride ion channels. So chloride ion is part of table salt, and it has to get in and out of cells, and it does so through little channels. And we recognized that these channels that they were studying were present on brain tumor cells, but not on normal brain cells. So we said, what if we put a little fluorescent molecule on it, and we allowed this little protein from the scorpion to deliver the fluorescent light into the cancer cells. Now this is what the molecule looks like. It's called a knotin. And you can see that it's kind of tied in a knot through these little bridges in the middle of it. And when we gave this molecule to a mouse that had a brain tumor, as you see on the right, there's a small tumor here, and you can see that the tumor paint that we made lit up that tumor beautifully, almost cell by cell, whereas in the mouse on the left, which doesn't have a tumor, you see no cancer. And so we're really excited this day to see that this pediatric brain tumor could be lit up by the scorpion molecule, bringing a fluorescent dye into the cancer cells. But I was also really worried that day because there are only a few hundred kids diagnosed each year with this kind of cancer, and we live in a capitalistic society. Which drug company is going to spend several $100 million or over $1 billion to develop a drug that would be useful for a few hundred children? Rather than trying to change capitalism, I think that the people who are CEOs of drug companies are so for a reason, and they would be irresponsible if they developed drugs under those circumstances. We decided instead that we would look to see, might there be a large group of people that would benefit from this. So I went around the Fred Hutchinson Cancer Center where I work, and I borrowed mice that had colon cancer and lung cancer and breast cancer and skin cancer. We tested tumor paint in them, and to our surprise, it worked in all of them. So we were delighted that day to learn that there's a path forward, and we started testing human tumors in these models, and we also saw that the tumor paint lit up those tumors. In one case, this prostate cancer mouse model, we'd already taken out the prostate cancer, and all these little red spots that are labeled with an N back here are lymph nodes that lit up, and each of those were packed with cancer cells. And the lymph nodes that didn't light up were not packed with cancer cells. But there's one spot that confused me, the spot right here, labeled with an L, and it didn't feel like a lymph node, they're rubbery. And this was just, it felt fatty to me. And we sent it down, and sure enough, it was a little wad of fat, but inside the wad of fat was a lymph channel. And inside the lymph channel were about 200 cancer cells that were crawling from one lymph node to the other. And so that day we learned that tumor paint was several thousand times more sensitive than an MRI, and that surgeons might be able to see it in real time while they're operating instead of looking at a scan that was taken 2 weeks ago that's stuck up on the wall. I think that a lot of people jump right from mice into humans when they're doing cancer work, and the failure rate in that is extraordinary. It's over 90% of the things that look good in mice fail in humans. We decided to take an intermediate step, and we looked at dogs, pet dogs that had any kind of cancer, whose families brought them in for care at the Washington State University Veterinary School. And we treated the dogs with cancer with tumor paint, and in this case you see that the tumor lit up, but the normal tissue around it didn't light up. And we moved to a dog that had breast cancer. Now, the surgeons knew that this was cancerous. They could feel that part. But again, if I gave you a scalpel, where would you cut? Typically, a surgeon would cut around this mass and would take a margin around it, send it down to pathology, and if the pathologist said the margins were clean, they would stop the surgery at that point and tell the person to get CT scans to see if there's any signs of disease in the future months. But this dog received tumor paint the day before the surgery, and what we found was that you could see that large tumor, but you could also see these extensions in other parts of the breast that were not visible to the surgeon. And so I think that this happens every day across the whole world, that people have a surgery for their cancer, they get the good news that their margins are negative, and then 6 months later, or 9 months later, or 2 years later, the cancer comes roaring back. And we'd like to make a change in that through the work that we do. This is a picture of the actual drug. How many of you take a drug that can help you find the toilet in the middle of the night? And I put this picture here because we've heard a little bit about funding from NHS and other organizations. When we had the data from tumor paint and we submitted grants to the National Cancer Institute, we didn't get any funding for it in the early days. They said, this is overly speculative, it may not work. And I actually wrote 6 grants, and all 6 grants were not funded. These little girls lost one of their best friends, Sydney, to brain cancer at age 14. In her honor, they started having bake sales and greeting card sales. And in the subsequent years, these 5 girls and the other kids in the neighborhood have raised $1.3 million to help us develop tumor paint. And they're part of a broader network of families that help us with fundraising to fill the gaps between what's available through the federal agencies. Now, we took that molecule and we transitioned it to a company called Blaze Bioscience that's now doing clinical trials. Tumor paint is now being studied in 5 human clinical trials. 46 patients have been dosed with it. Because it's being evaluated by the Food and Drug Administration, I'm not able to tell you the efficacy and the safety data on it, but some of that information will be out in the next couple of weeks, so some of you will be able to follow us on our website and see what it's starting to look like. But as we transfer that out of my laboratory and into Blaze, I had an opportunity to completely redevelop my lab and asked, what do we want to do? And so again, I thought about these kids that I take care of, and each week when I go to clinic, I might meet a child like Kyle, and the very first day that I meet him, I have to tell his parents and his family that he probably won't be around in a year because of the type of cancer that he has. So we wanted to go after those kinds of cancers that are not surgically removable, and we wanted to go after other diseases that are incurable. And we had this whole decade of experience of working on the scorpion molecule, which is a very unique little mini protein that I thought really could potentially provide a template for other drugs, for other diseases, like the one that Kyle had. Now, these molecules that are found in nature as drugs are called knot molecules because they're tied in knots. And about 300 of these have been identified by other scientists around the world in the previous 23 years. Plants and animals can't go to Boots to get their drugs that they need to get through the day, but they need drugs in order to survive and in order to reproduce. And in addition to the scorpion example that I gave you before, the example on the right here is the Brazian fruit in Africa. Anybody understand why fruit is sweet, why we enjoy eating fruit? Fruit is sweet because the plants that make the fruit want animals to eat it and spread the seed. Okay, so it makes sense, right? That's the reason that fruit is sweet. Well, the Brazian tree, rather than spending energy to make sugar, makes these little mini knotted proteins that bind to the sweet taste bud receptors on our tongue, and they're 1,000 times sweeter than sugar, and they don't have to expend all that energy, and it's a way that they get their seed spread without using as much energy as other plants. I could give you dozens of examples of how plants and animals use these little knotted proteins to create their own drugs. There are 2 molecules that have been approved by the US Food and Drug Administration that came from plants or animals that have this little knotted protein sequence. One of them comes from E. coli, which, as you know, causes diarrhea, and it does so through one of these little knotted proteins that binds to a receptor inside the gut. And so this is used as an anti-constipation medicine. On the right is the cone snail. The cone snail sits on the bottom of the ocean, but it catches fish. How does that happen? It has a little stinger that comes up and injects the molecule you see on the right into the fish and paralyzes them. They float down and the cone snail eats it. This is being used to control pain. It's injected into the cerebrospinal fluid for people that have chronic pain, and it's able to relieve their pain. Well, these are two nice examples of gnatins being used as medicines, but the issue here is that the exact molecule that was used by the plant or animal served the same purpose in humans. Now, if we want to try to cure cancer or Alzheimer's disease or autism, there is no basis in plants or animals to try to create drugs that would do those things. So we're going to have to make hundreds of thousands or millions of variations of these in order to create candidate drugs for humans. The scientists that have been working on this previously had identified 300 over 23 years, and my team identified 206,000 more in a day and a half. The reason we did that is because a lot of things that when I mentor young students coming up in science, I always tell them, don't do things the way your mentor did them. Do things the way you would do them in 2015. And this guy came from Oracle. He had written a Python program, and he just went at it and found all these computationally without ever touching a scorpion or a spider or a sea slug. Now these knot and peptides this is the structure of them. This is how they tie themselves in the knot. And when we change them, we call them optides, for optimized peptides. In the past, people had to get these molecules from animals literally by milking scorpions or by making them in the lab. When I first made them in the lab in a sabbatical in Australia, we could make about 1 molecule a month. And if you need hundreds of thousands or millions of these, that's going to be a long road, right? So our team learned that we could computationally design these molecules. And furthermore, we could order genes online now. So my team can design these on the computer, type the code in for what we want, give them my credit card, and the FedEx guy comes on Tuesday with a little packet that has 10,000 genes in it. My team then takes those 10,000 genes and we put them into a virus that's a modified HIV virus that can no longer cause AIDS, but it can take that DNA and squirt it into human embryonic kidney cells that have been immortalized. These human embryonic kidney cells then become factories that produce the drugs. They pump it out all night long. My team comes in the morning and begins purifying the drugs. We've been able to create this little factory to make these, and we can make large volumes of these, enough that we can do animal studies. Other companies that make these types of molecules, it takes about $5 million and about 9 months before they can do their first animal study. For us, it's about $3,000 in 3 weeks. Furthermore, we've learned how to roboticize this so that we can make hundreds of these molecules a week, and we just ordered the first robot that's coming in a few weeks. We've learned how to flip pharmacology on its head. We've learned how to learn from these molecules where they go in the body, and then how to use those as templates for diseases that affect those areas. Here you can see that we've identified molecules that go into the brain that could be useful for autism or Alzheimer's disease. And we've even found one molecule that bathes the new neural stem cells in the brain, which are the ones that are being created, and those are the ones that would be most useful for people with neurodegenerative disease. We were very surprised to find 4 molecules from scorpions and spiders that all only go to the cartilage in the body and almost nowhere else. So if any of you have ever had a sore hip joint or a knee joint or any kind of arthritis anywhere in your body, one of the things that we're doing for you is to create a molecule where we can deliver the pain-relieving medicines or the joint-restoring medicines directly to your joints and bypass the rest of the body so you don't have the side effects of systemic steroids or other medicines. A lot of times people ask me the question of, if you can deliver light to the cancer, why couldn't you deliver chemotherapy to the cancer? And we're in the process of doing that. Here we found a molecule that goes just to the tumor, but not to the liver, the spleen, or the lungs. And this molecule we're developing as a molecule that would deliver chemotherapy right to the cancer. And this molecule came from this little guy, the grasshopper. And finally, we've identified through whole genomic studies new targets in brain cancer that, even though they're not mutated in those cancers, are very vulnerable in those cancers. And in this case, the BUB1B, when we knock it down in normal cells, the cells go through division properly. But when we knock it down in brain tumor cells, those cells explode. They go through mitotic catastrophe, and that's exactly what I want to happen to cancer cells from what I do taking care of these little kids each week. This is the team that's doing the work. Interestingly, when I began this project, we had 2 scientists working on the project. I announced in June of 2013 that we're going to start a new fundraising effort to help build the team, because what I didn't want to do was spin this out into a biotech company that would necessarily have to sell the whole company to a pharmaceutical company And then all these libraries of drugs would end up in the hands of one decision-making committee that's driven by profit. And so we decided that we're going to crowdfund this effort to the best of our abilities. And I was taking care of a little girl then whose name was Violet, and she passed away from her brain tumor. But before she passed away, she asked if we would do an autopsy when she died and take her brain tumor and make tissue to share with other scientists around the world. And I thought that was so generous that we named this Project Violet after her. And we began raising the funds through this online source. In the past few years, we've raised over $8 million through Project Violet and related fundraising efforts, and have built the team up to 34 scientists. So we're now in the process of creating these drugs for other incurable diseases. It's become a worldwide effort. I encourage you to follow us on Project Violet Facebook. And I thank you very much for your attention and for a wonderful day.